My name is Megara Ramirez. My science project this year is how do scientists make our DNA?
What is our DNA?
Well, our DNA is two pieces of DNA together. DNA is a book that's in your body that tells cells what to do. It's about 2 meters long. There's about 2 meters amount of DNA in one cell. We are going to figure out how to cut and paste DNA. We are going to find the cutters to do it. The cutters are a certain enzyme that they search the DNA strand for a certain piece to cut. There's another enzyme that can paste them. The one that cuts them leaves sticky ends. The one that pastes them, connects them together.
Now, is there, when it cuts it, does it cut it all the way through? I mean, where does it actually cut it at?
At the sugar backbone. They cut it at the sugar backbone. DNA is being like a ladder. You don't need to cut what's in the middle to cut it in half. You must cut the sides.
Okay. Now show us what you're going to do.
Well, what we're going to do is we are going to go to generate a set of DNA. Then we are going to highlight it. See. Then we are going to click Control Copy. Then we are going to click the number that it is on and highlight it. Then we're going to click Control Paste, and it will paste the DNA sequence. Then we go on to the other side.
What is this other side for?
The other side is the neb cutter.
What's a neb cutter?
The part that cuts.
Okay.
We click the box and we click Control Paste. You must click it and then click it while doing it, or I'll just do V. Then you do Submit. Then,
wait a minute, what is that?
This is the DNA. Each of these are enzymes that will cut it.
What's the red line?
The red line is where it will cut. And this one it will cut right there. This one will cut the same place. And these do the same thing. It will go all through the entire thing.
Those are sticky ends, right?
The little dots are sticky ends.
So now my question to you is can you put any sticky ends together?
can't put any sticky ends together. You can only put A's to T's and C's to D's. Then we click print. Then we do map only and create image. Then we click here to view, download the PDF file. Then we click down here under there's print. Then we click print again. And then we wait for the DNA sequence to print. Next, you must write the number on it.
What's the number for?
The number is so you don't get confused with all of them. Number 15 is this one.
Now what are you going to do once you get them all done?
We are going to compare the enzymes that cut and paste them. That way we can see if there's any enzymes that can cut two. That can cut almost all of them.
So you're going to see what enzymes cut what, where, right?
Uh-huh.
What do you think about your science project?
I'm really excited about it.
So I guess we'll come back when you actually compare them, or do you just want to videotape this part?
We have to compare them.
Okay, so I guess goodbye for right now?
Yes, goodbye. Hello again. My name is Megara. The name of my science project is Poetic Enzymes. I named it that because DNA is like a book, and the enzymes that cut the DNA so it's like poems. My question was, how do scientists change DNA? My hypothesis was, I think the scientists put DNA in a dish and mix a solid with it, like I did when I used salt to take DNA out of fruit to cut it up. Then they mix the pieces with other DNA, and they have different DNA. My hypothesis was wrong. To figure that out, we went onto the computer and got DNA maps. Then we took every single enzyme that's on it and put it here.
Can you show me the maps?
Like this one, this one, this one.
Can you lift them up so I can see them? Okay. How many of them did you do?
20. We only write each enzyme down once. Then we made a tally chart. Put every single enzyme down in alphabetical order. And And we looked in here and saw every single one that we saw. We marked down one line. Then, if there's only one enzyme, if that enzyme only did it in one, it will have a pink one. If it did it more than once, it'll have a purple one. Then we marked out how many there is here.
Let me see.
We marked out every single one that was right here. That was here. We did it the same way as here.
You colored it to match?
Uh-huh. The enzyme over here. Then we did this. There is 7 common ones on this one and 3 unique ones on it. Then we took the numbers that are right there. Oh. I'm sorry. And put them down right here. This chart was here in the last video. These are all the DNA. These are all the DNA strands. The purple ones. The purple one. The purple one is common and the pink one is unique.
So how do they make DNA? How do they change it?
The DNA enzyme reads the DNA until it finds the right spot to cut it at. RLAL is the only one in all of them. There are enzymes that can cut in more places. Like there is an RLAL that cuts GG and an RLAL that cuts GT.
What did you learn about the enzymes?
I learned how they cut it.
Did you learn about whether they're unique or common and what's the difference?
Yes. If it was only one, you marked it pink. If it was more than one, it was marked purple.
What's pink and purple mean?
Pink is unique and purple is common.
What does that mean?
It's how many there is.
How many times it's used?
Yes.
Now, what else did you learn?
The RDNA is made by an enzyme that cuts the DNA strand. It only cuts the sugar phosphate backbones.
What does it make when it cuts it?
It leaves sticky ends.
Now the when they
And then they put another piece of DNA in with it.
any kind of DNA?
No, only the one that will match the sticky ends. The A to C or C to G.
Now what do they do?
Then they make it. Then they put an enzyme that will put it back together. And put it in with the two DNAs. It'll connect the sticky ends on the sugar phosphate backbone.
After you have the rDNA, what do?
Then you copy it.
How do you copy it?
Well, before there was other ways to copy it, you had to put it in a cell. To put it in a cell, you must put the DNA in with the cell in the test tube. Then you make it really cold.
Well, why won't the DNA just go into the cell?
Because they're both negatives. Calcium chloride is positive. It makes the DNA and the cell neutral.
Then what do?
Then they make it really cold, so the inside is cold. Make it really hot, so the outside is hot.
The outside of what?
The outside of the cell
So the inside of the cell is...
cold and the outside is hot.
Then what happens?
Then the bacteria sucks the DNA in to get the warmth then through the little holes in it.
Okay. Then what does it do?
Then the cell copies it.
So the cell does the copying?
There are now other ways.
What are the other ways?
One of the other ways is PCR.
What is PCR?
Denaturing, annealing, and extending.
So what's denaturing?
Denaturing is when it splits it apart.
Splits what apart?
Splits the DNA apart.
And how do they do that?
They make it really hot for the DNA apart.
Then what happens?
Then they will put anneal it,
How does that work?
putting a little piece of DNA on with it.
And what does that little piece of DNA do?
It finds the spot where it is supposed to, that matches it. Then the extending happens. They
But when they...
put an enzyme that will extend that piece.
Okay, then what do?
Then they make it denature a nail and a stem over and over again.
Now, is there something that has to do with warming it up and cooling it off?
Yes. When it cools off,
when they denature it, they warm it up, right?
yes,
And when they nail it,
they cooled it off.
And then when they extend it, they
don't change the temperature at all.
Okay. Anything else you've learned?
I have learned that RLA is in every single DNA strand.
Have you learned any ways that this technology is used?
Yes.
How is it used?
In DNA tips and microarrays.
What's a DNA chip?
A DNA tip is where they have DNA on a little piece of glass. Then they put a certain light on. It'll take away the layer that protects from anything joining it. Then they put another one. Then they put an A's, T's, or C's on. Then they sprinkle it on.
What about G's?
A's, C's, T's, and D's.
Okay. Then what do they do?
Then they do it all over again. There are little stools that will help. They sign it through the stool.
Stool or filter?
Filter.
Then what do they do after they finish putting the DNA on there?
Then they do it over and over again. And they'll...
Do they put a cover on it?
Yes, they'll put it in a case and they'll label it. You don't want to get mixed up with them.
What about the
microwaves?
Yes.
It dips pen-like points into little DNA pieces.
Pieces or little cups?
Cups. And it will basically print onto the
They print it on paper?
No. They do it on a piece of plastic.
Plastic or glass?
Oh, glass. And it is broken up into little squares, the same size.
Now what about medicines?
DNA is also used in medicines such as cancer medicines, diabetes medicines, and reduction medicines.
Now how do they get the diabetes medicines? How did they do that? How did they do it?
They use a machine to do it.
Do they use it in any way, the way they do how you make the rDNA and then they put it into a cell and the cell copies it over and over again? Is that how they do it? Do you remember?
Not really. All I remember is they use a machine.
What are the other kinds of medicines?
They do it as such as diabetes medicines, cancer medicines, and reduction medicines. Reduction medicines are used so that the body doesn't reject organs and pieces of the body that other people donate to them. For my cousin Gregory. When he was in his mom's tummy, they got into a car accident and one of his livers got hurt.
Livers or kidneys?
Kidneys. I mean. Since you don't have a Zach and his mom donated part of her liver.
Kidney?
Yeah, kidney. Part of her kidney for him. Since they don't have the same DNA, well, Gregory's body tries to reject the piece of kidney. So they give a medicine to him to make his body so that it doesn't reject the kidney.
What did you think about your science project?
I like my science project.
Do you want to keep learning about DNA? Anything else you want to say?
There is so much more that I don't know.
You want to learn?
Yes.
You want to say goodbye?
Bye.